Hey, what's up, everybody? We are going to begin our second video on the brain today. We're going to wrap up our lobes of the cerebrum, starting with the occipital lobe, light blue all the way in the back here, posterior to the parietal and temporal lobe. There is a parieto-occipital sulcus separating the parietal and occipital lobe as well. Now, my occipital lobe has two areas that I am worried about and interested in. Sorry for the noise, just adjusting my headset there. So, in my occipital lobe, two major things I'm worrying about are my primary visual cortex as well as my visual association area. And we've seen this situation before with a primary and an association area. Now, the primary visual cortex is all the way in the posterior most part of the occipital lobe. It is all the way back there. It's actually a good sized sensory cortex. It's where we ultimately receive input from those light receptors, which are called rods and cones, in the retina of our eye. So we know signals come out of the retina via the optic nerve, optic chiasm, optic tract, to the thalamus, then onward to the occipital lobe, and it's to the primary visual cortex where that information is going. Now, one of the things about the pathway of vision is it's sort of contralateral, but also sort of ipsilateral. So, like, if I'm looking, I got like a superior view here. Here is my right eye. On my right eye, some of the fibers are ipsilateral, and then some of the fibers are contralateral. So the nasal fibers end up being contralateral, nasal fibers here being contralateral. Then the more lateral temporal fibers end up being ipsilateral on both sides. Now, what this ends up doing is creating a situation where my visual field, like imagine what you're looking at, like the entire breadth of the view in front of you, and your right visual field, all that information ends up going to your left occipital lobe. Then your left visual field, all that information ends up going to your right occipital lobe. So it is contralateral in the sense of the visual field, but as far as each eyeball goes, each eye is going to be both ipsilateral and contralateral. So check out how the optic nerve, like if I cut this optic nerve, well, I'm not going to see out of this right eye. If I cut this optic tract, that's going to impact both eyes, which is pretty interesting, pretty neat. Okay, I see that in just another diagram here. Eyeball, fibers in the optic nerve. The medial ones are switching sides, the lateral ones are staying put. Optic tracts projecting to the thalamus, thalamus then sending signals to that primary visual cortex, which we see back here. Okay, the visual association area is in front, anterior to the primary visual cortex. And the visual association area is involved in the processing, recognizing, understanding, remembering visual stimuli. And we saw similar things in this other association areas, like the somatic sensory one and the auditory one. So same basic idea. And now we got the occipital lobe down. We got one more lobe to do, which is the insula. The word insula means island because this lobe is deep within the lateral sulcus. It's kind of hidden and separate from the other lobes. Now, my insula's got two important parts that I'm worried about. It's got what's called my primary gustatory cortex as well as my interoceptive area. There's our insula again right there. My primary gustatory cortex is all about taste. The word gustatory has to do with taste. So taste receptors in the tongue are projecting their information ultimately to the gustatory cortex in the insula. Now, interoception is sensation of the body's viscera, and your insula is involved in that. And by viscera, I mean internal organs. So, for example, this is the stomach right here. Of course, you can tell when your stomach is stretched and full. Well, your insula is involved in that. This is the urinary bladder. This is another organ that we can, of course, are going to be familiar with when it's stretched. So 2 examples of interoception where we're aware of internal organs, aware of viscera. And with that, we are now done with our discussion of the separate lobes. I want to talk still a little bit about the cerebrum because we've got cerebral nuclei. So we're going to have, here we are, some cerebral nuclei. Subcortical, so deep to the cortex, clusters of somata, in other words, gray matter. So, for example, we've got a bunch of cerebral nuclei labeled here. I'm not going to make you know all these names, but you should know that cerebral nuclei are involved in movement, learned behaviors, emotions, and moods. And by the way, cerebral nuclei, I think we mentioned this in the last lecture, they're also called basal nuclei because they're deep, they're not superficial. All right, let's finish up our cerebral discussion with taking a look at the 3 basic types of white matter. Now, we know all white matter is made of myelinated axons, which is sending signals from one part of the brain to another. And there are actually 3 basic types of cerebral white matter. There are association fibers, which are going to connect different parts of the same hemisphere to each other. Then there are commissural fibers, which connect one hemisphere to another. The word commissure means adjoining or connecting. Then finally, there are projection fibers. Projection fibers are going to connect the cerebrum to other parts of the brain, like, for example, the cerebellum or the brainstem, Diencephalon, or to the spinal cord itself. So those are my 3 basic types of white matter fibers I'm going to find in my cerebrum. If I look at this picture here, it's just kind of giving me a nice little colorful diagram. So association fibers are staying in the same hemisphere. Commissural fibers are going from the left to the right hemisphere, from right to left. And projection fibers are going to leave the cerebral hemisphere, going to another brain region, maybe the brainstem, or maybe even onward to the spinal cord. So those are my 3 basic types of fibers. If I look at this picture, again, I got some nice colorful examples, like these pink guys here, staying within one hemisphere. These are association fibers. The green going across, those are commissural fibers. And our best example of a commissural fiber structure is the corpus callosum, which we definitely saw in lab, brain model, sheep brain. There are other commissural fibers as well. This is not the only set. Then finally, I got some projection fibers. I see some going to my diencephalon. I see others going down into the brainstem. Continuing onward as well towards that cord. Just another view here. So what kind of fibers do I have right here? These would be commissural. And then coming down right here, these would be projection. So that's pretty much my overview of the white matter in my cerebrum. And there, I forgot I got one more picture. This is a view of the fibers of the corpus callosum. You can just see how crazy this is. So we're kind of like pulling the 2 hemispheres apart, like opening the longitudinal fissure, so we can really look in there and see all the fibers of the corpus callosum. Okay, we're done with the cerebrum. Yes, we've only got 3 more brain regions to do. We got to do the Diencephalon, which is what we're going to start now. And then we're going to knock out the brainstem. Then we're going to knock out the cerebellum. And then we'll talk about brain protection. Then we'll be done with the brain. Now, my Diencephalon forms the central core of my brain. It is going to consist of 3 gray matter structures, including my thalamus, my hypothalamus, as well as something called my epithalamus. That is something we did not meet in lab, the epithalamus. Although, as we'll see, we kind of did. So I can see my Diencephalon right here, covered up by my cerebrum. If I slice my brain mid-sagittally, so this entire area, haven't busted out my drawing tool recently. I know you guys appreciate all my artwork, but we've got the Diencephalon as a whole right here. Then there is our thalamus, our hypothalamus, and the epithalamus is a couple different structures, including the pineal gland, which we did meet. So the pineal gland, which we met in lab, we now know it's part of something larger called the epithalamus. Okay, let's talk about the thalamus first. No, let's not. Let's look at all 3 of them again. Sometimes I forget that I got another slide. I'm pretty good with knowing like what's next, but occasionally I forget. All right, let's talk about the thalamus. So if we look in this brain here, we see like these 2 eggs here. These 2 eggs are the thalamus. So they're actually pretty big, bigger than they appear in a mid-sagittal section. About 80% of the Diencephalon is actually made up of the thalamus. And as you can tell here, there's a right and there's a left thalamus. And those 2 halves are connected by this little bridge here, which is called the interthalamic adhesion. Now, my main job of my thalamus is to be a sensory relay station. It is going to edit, sort, and route sensory information coming in. And in fact, all of my senses except for one go to the thalamus before they go to their appropriate cortices. So the only one that doesn't go to the thalamus is my olfactory sense. It goes right to the olfactory cortex. But visual signals, thalamus first. Gustatory signals, taste from the tongue, thalamus first. Hearing, thalamus first. Somatosensation, thalamus first. Balance, thalamus first. Proprioception, thalamus first. All right, so the main job of the thalamus is to like help us ignore things that don't matter, because it's a tremendous number of signals, tremendous amount of information we're getting all the time. And so we obviously have got to filter some of it out and only allow things that are really important to make their way onward to the appropriate cortexes. Cortices is actually the plural, not cortexes. Okay. Thalamus does have cognitive and motor roles as well, but primarily it is that sensory relay station. Now the hypothalamus is inferior to the thalamus. So here's my hypothalamus right here. There are a lot of named nuclei in the hypothalamus. We're not going to learn all of its names, all of their names, but we are going to go over some general functions of the hypothalamus. And notice how the pituitary gland is attached to the hypothalamus by what's called the infundibulum. Okay, a lot of functions in my hypothalamus. Thermoregulation. So it's involved in maintaining body temperature. It makes and releases certain hormones. It is going to be involved in my sleep-wake cycles. Appetite, satiety, memory, learning, wakefulness, sexual arousal, blood pressure control, heart rate control, gastrointestinal control, thirstiness. And this isn't even all of them. Like, this is a pretty darn good big list, but it is not all. It's not even all of them, which is the amazing thing about the hypothalamus. Um, so that's pretty much it for our hypothalamus, except for its attachment, which is the pituitary gland. So my pituitary gland is attached to my hypothalamus via this little stalk of nervous tissue called the infundibulum. Now, my pituitary gland actually has two very different regions. There's an anterior pituitary. The anterior pituitary is known as the adenohypophysis. Then there's a posterior pituitary, which is better known as the neurohypophysis. Nice, good words there. I like these a lot. Now, my adenohypophysis, which is my anterior pituitary, is going to release several hormones, including some reproductive ones like follicle stimulating hormone and luteinizing hormone, as well as one involved in milk production, prolactin, one involved in the growth of muscles and bones, which is growth hormone, and there are even more. So my adenohypophysis makes and releases these. Now, follicle stimulating hormone and luteinizing hormone, like I just said, they're both reproductive hormones, so they affect the production of other hormones by the ovaries and the testes. They also help promote the formation of gametes, which are sex cells like spermatozoa and oocytes in the testes and ovaries, respectively. Now, prolactin is involved in the production of milk from the mammary glands, also involved in sperm production in boys, and it's got some other reproductive roles as well. Okay. Growth hormone causes growth in many tissues, actually increases your muscle mass. It helps increase bone growth. When you've still got that epiphyseal plate, it'll make your long bones longer. Also helps decrease your adipose tissue levels. So that's just a few facts about growth hormone. It is not a complete list here, of course. Now, meanwhile, my neurohypophysis actually doesn't make hormones on its own. My neurohypophysis is going to get hormones from my hypothalamus. So there are going to be neurons in the hypothalamus that are making hormones, and those hormones travel down axons into the posterior pituitary. So neurons up in the hypothalamus have axons running through the infundibulum and then ending in the posterior pituitary. And the posterior pituitary is basically like storing those hormones, then eventually releasing them. And the two major hormones that are doing this are antidiuretic hormone, which is also called vasopressin, and a hormone we met in the first video we did this semester, which is oxytocin. So let's talk about those for a moment. Antidiuretic hormone, aka vasopressin, it decreases urine production in the kidney. Antidiuretic means against peeing, so it's going to tell the kidney not to make as much urine. If we make less urine, that raises our blood volume and blood pressure. Meanwhile, ADH, antidiuretic hormone, it's also called vasopressin. Vaso means blood vessels. So vasopressin, it's going to cause the blood vessels to contract. The smooth muscle in arteries is going to contract. And that also raises blood pressure. Okay, cool. Oxytocin causes smooth muscle contraction in the uterus during childbirth and causes smooth muscle contraction in the mammary glands to move the milk during lactation. And oxytocin has roles in boys as well, especially in bonding, that sort of thing. Okay, awesome. done those hormones. Now the epithalamus is the posterior section of the diencephalon, and my epithalamus has a couple different parts, but the only one we're going to talk about is the pineal gland. And there's the pineal gland sitting right there. The pineal gland, of course, we met it in the lab. The pineal gland makes this hormone called melatonin. So actually the absence of light hitting your eyes promotes melatonin production, and melatonin is going to help modulate sleep patterns, sleep-wake cycles, that sort of thing. So that's the pineal gland. Let's do the brainstem real quick. Then we'll quit this video here. I was kind of debating, do I want to stop here? Do I want to keep going? So the brainstem is inferior to the diencephalon. It's got three major parts. We got the midbrain, then we got the pons, then we got the medulla oblongata. The midbrain is that first part, just inferior to the diencephalon, superior to the pons here. There's a passageway running through the midbrain. This is called the cerebral aqueduct, which is going to link my third ventricle, which is a cavity up here, to my fourth ventricle, which is a cavity down here. Now, in the front of the midbrain, we've got these structures here that bulge. They're called the cerebral peduncles. So this right here, this collection, you can see it looks like it's a collection of bundles of axons, and this is the cerebral peduncle. It's got descending motor fibers and ascending sensory fibers. By the way, I love this picture because check out that thalamus. Look at that right thalamus right there. And there's the left over there. You can appreciate the size of the thalami in here. There's the pineal gland, by the way, hanging out right there. Awesome. Now, posteriorly, my region behind the aqueduct is called the tectum. In the tectum, in the back of the tectum, we have four bulging structures. 1, 2, 3, 4. And those are my colliculi, and that group of four is called my corpora quadrigemina. Quad means four, gemina means look-alike, corpora means bodies. So my top two parts of my corpora quadrigemina are my two superior colliculi. And so then, of course, my bottom two are going to be my inferior colliculi. The word colliculus, by the way, just means little hill. So because it's a bulge, it's a little hill. Okay? Now, my superior colliculi are involved in a variety of visual processes and visual reflexes. Often involving in, like, keeping your eyes oriented to a particular thing happening. Like, I always use the example of catching a baseball, of coordinating your eyeballs, your body, and your hands to do that. I'm actually going to skip the defining of these three here and leave our superior colliculi function at what I just said. And there it is on the sheep brain. Look at that right there. So my inferior guys are my inferior colliculi, and those, of course, are involved in auditory reflexes. There they are on the sheep brain right there. Let's just do one of these, the startle reflex. So the turning of your head towards a loud sound, that is your startle reflex, and that is mediated by your inferior colliculi. It's a nice brain picture. I see my inferior colliculi. I see my superior colliculi. I see the pineal gland. I see a nice cerebellum with a vermis in the middle. Beautiful. All right, that rat is turning towards a loud sound. His superior colliculus is, his inferior colliculus. This video is going on too long. I'm starting to make mistakes. His inferior colliculus. Okay, the pons is below the midbrain, above the medulla oblongata. So there's the pons, there's the midbrain, there's the medulla, and the pons is anterior to the good old cerebellum. Pons has a variety of nuclei in it, involved in things like respiration, involved in peeing, sleeping, swallowing, and more. The word pons means bridge because there is a lot of white matter in the pons, taking sensory information up and motor information down. Okay, done the pons. The medulla oblongata is my final inferior part of my brain stem. And it is, of course, superior to my spinal cord, which we see down there. Medulla oblongata also has a lot of motor and sensory fibers in it. It's got nuclei involved in blood pressure control, heart rate control, breathing, vomiting, sneezing, burping, and more. So if I look at my anterior medulla oblongata, I do see these two ridges here, these two bulges here. Each of these is called a pyramid, and these contain the axons of those upper motor neurons from my primary motor cortex, which is my pre-central gyrus. And so those neurons were called pyramidal neurons, and these are their axons, and they're called pyramids. Now, it's in the pyramids that we do the switching of sides. We can kind of see that. The little crisscrosses that are going on right there. So, axons from the left primary motor cortex are coming down in the medulla and switching sides. And ditto on the right. And this switching of sides is what results in the contralateral motor control. The fact that your right hemisphere controls your left side and your left hemisphere controls your right side. The switching is called the decussation of the pyramids. The word decussation more or less means to make an X. And because we see like little X's right here, that's where the word comes from. By the way, check out the thalamus up here. Look at that thalamus. Look at that pons. Look at those cerebral peduncles of the midbrain. Look at that infundibulum right there. Look at those mammillary bodies. Look at that optic chiasm, optic nerves right there. Right there. Look at that optic tract, other optic tract. Look at that trigeminal nerve. Look at that other trigeminal nerve. All sorts of good stuff going on in this picture. Sweet. All right, we're calling it quits. We did the brainstem, we did the diencephalon. We're going to do one more brain video on the cerebellum, then on the protection of the brain. So bone, meninges, CSF, blood-brain barrier, those sorts of things. And that should be a nice quick one. This one wasn't too bad, under half an hour. As always, let me know if you have questions, and I will see you later. Bye-bye.